introduction in chapter 10 was some things that you needed to know in order to understand photosynthesis better. So you should have a grasp on the chloroplast anatomy. So what does that mean? That means it has two membranes. It has the thylakoids that are all stacked up to create what we call a stack of thylakoids. Grana. Yep. And the fluid inside the chloroplast is the stroma. All right. So that's what I mean. So I'm not going to draw that again and so on. We've done that before, but you should know that. You should also know how the reactants get to the chloroplast. So to answer this, you need to know what the reactants are in photosynthesis. So what are the two main types of molecules that are needed for photosynthesis to happen? Carbon dioxide and water. So carbon dioxide, how does carbon dioxide get into a plant to do photosynthesis?
Is it the stomata on the bottom of the leaves?
Yeah, absolutely. The stomata at the bottom of the leaf, remember they open and close and allow gas exchange to occur. And then how does water get in? Roots, right? And then light is also needed. It's not considered a reactant, it's not a physical substance, but light is also needed as an energy source. What absorbs the light? Pigments in the chloroplast, right? Mainly in the leaves, the cells of the leaves. All right, so that's that. You should also know the equation, so that would be the reactants and the products, and kind of the overview. So know the equation. So and what I mean by overview is that there are two main phases of photosynthesis. What are the two main phases? One starts with the L. Light reactions, right? And then after the light reactions becomes the Calvin cycle. And if we were to think of the big picture as far as what's the light reactions, what's the purpose of the light reactions? It's to make something. Actually, two things.
ATP and electron carrier?
ATP and the electron carrier called
NAD.
NADPH, correct? All right. Why these are important is they are used in the Calvin cycle. So that's the only purpose. So the ATP made in the light reactions was not used for cellular respiration and so on. It's going to be used directly in the Calvin cycle. And at the end of the Calvin cycle, what does the Calvin cycle make? At the very end, it makes glucose. It first makes G3P, which is a 3-carbon molecule, and then two of those make a glucose molecule. 6-carbon. The other thing is, with the equation, what goes in and what goes out, that you need to be familiar with is tracking atoms, meaning when we talk about water and CO2 going in, and you get glucose and oxygen out, where do those atoms go? Where does the carbon, so in carbon dioxide, come from? Well, it obviously comes from carbon and glucose come from. It comes from carbon dioxide. It's the only reactant with it comes from the CO2. So I had color-coded the notes and so on, so just take a look at that and review that. And then review about light energy. So we talked about the electromagnetic spectrum, and that the electromagnetic spectrum, that there's a part of that called that's visible light, that is useful for photosynthesis, and that each one of these sources of radiation have different wavelengths. We talked about that, wavelengths of light and what that means, and so on. So some just basic review about light energy and so on. So this is kind of the big overview and things that you need to know to understand better photosynthesis. So now the rest of the chapter was about the light reactions and then the Calvin cycle. So let's look at the light reactions first. So the light reactions are called that because you need light to happen. We just said that what absorbs light? Pigments. So, anybody remember what the main pigment in photosynthesis is? Chlorophyll A. So chlorophyll A is the main pigment. A. I was thinking A already. I'm writing that. Chlorophyll A. Are there any other pigments involved in photosynthesis?
Yep.
Yeah. Collectively, these pigments are called accessory pigments. Accessory pigments are things like chlorophyll B, carotenoids. You don't have to memorize or know all the different wavelengths of light that all these accessory pigments need, but you need to know what the purpose of accessory pigments are. Why is it that there's just not chlorophyll A? Why are there accessory pigments? What do? They absorb different wavelengths of light. So it allows the plant to absorb lots of different wavelengths of light from the visible light, versus if you just had chlorophyll A, they would just absorb one wavelength of light. And so therefore they get more energy ultimately to do photosynthesis by having the accessory pigments. Okay? So then we studied pigments, and scientists have studied pigments and figured out what pigments either absorb and reflect different wavelengths of light. So if it's a photosynthetic pigment, the wavelengths of light that it absorbs is the wavelengths of light that are useful for photosynthesis. If it doesn't absorb the light, then it can't be useful for photosynthesis. So they use a machine called a spectrophotometer to measure what goes on with pigment. So if you recall that you can put a little test tube in a machine that has a solution of a pigment. So this is the pigment in a little test tube. And then you can pick a certain wavelength, that's the shorthand for wavelength, certain wavelength of light. So you can pick whatever wavelength you want, you beam it at the pigment, and then on the opposite side it measures the percent, what's called the percent transmittance, how much light is transmitted. And so if you have a high transmittance, what does that mean in terms of absorbance? What's the relationship? High transmittance means low absorbance. So that's the relationship here. High transmittance equals low absorbance. So then, why is this important? Because if this is a pigment from a plant, and we want to see, well, what wavelength of light does this pigment absorb, we can figure that out by beaming the different wavelengths of light and figuring out the percent transmittance. And so there's a very high transmittance that that means they're not absorbing that particular wavelength of light. But if there's a very low transmittance, not very much is going through, that means the pigment is absorbing that wavelength of light, and that wavelength of light is probably important for photosynthesis. Okay? So so then in studying what wavelengths of light are important for photosynthesis, let's look at this, wavelengths, chlorophyll uses. So what they did is create two things, two graphs. One's called an absorption spectrum. And the second is called an action spectrum. And these they're both in the notes, they're also in the book here. But like the absorption spectrum, what it looks like is that you had your wavelengths of light here, so all your different nanometers or wavelengths of light, and then they absorbed what the absorbance of, when you beam the light, like this spectrophotometer, what's the absorbance of that. And so you can take different pigments like chlorophyll A and see where there's a high absorbance. And so remember there was a graph that looked like this, and it went down like this, and it went up like that. Do you remember that? And so if you have areas where there are peaks or higher absorbance, then we can make the hypothesis then that for that particular pigment, since that particular pigment is absorbing that wavelength of light, then it's probably important for photosynthesis. That's what an absorption spectrum is. An action spectrum looks very much the same. It has the wavelength of light, but instead of absorbance, this is the rate of photosynthesis. So how much photosynthesis is going on. And so you can see here where you have the peaks of absorbance, you would expect peaks of photosynthesis where there's high absorbance. Does that make sense? Okay? And so that's the action spectrum. And so there's an experiment with this. A man named He came up with the action spectrum by using a thing of algae and bacteria, and the bacteria grew in the shape of the action spectrum. So review that. So let's look at then what actually goes on in the light reaction. So obviously light is absorbed and by the pigments, and so where they're absorbed by the pigments is in a photosystem. So let's go to the next page here. So let's review what a photosystem is. Two parts to a photosystem. Light harvesting complex. And the second thing is called the reaction center. The reaction center has two parts to it. This is where the chlorophyll A is, the main pigment molecule, and what's called the primary electron acceptor. So in our notes and in the book, it looks like this. So this whole thing is the photosystem. This part right here, outside, the outside part is called the light harvesting complex. And your book has just a little kind of rectangle at the center of it. That's the reaction center. And the reaction center has the chlorophyll A, chloro A here. And I'll just put PEA for primary electron acceptor. So that's kind of the what it looks like there. On the light harvesting complex on the outside has these little molecules in them. It's called light harvesting complex. What do you think these molecules are? What harvests light or absorbs light? The different types of pigments. And so these little green dots represent pigments. So what do pigments do? They absorb light. So here's your light energy. I'll just use it in orange. Here's your light energy. Your pigments absorb the light, and then the light gets transferred from pigment molecule to pigment molecule. Eventually, all the light energy that's absorbed by all these different pigments get transferred to chlorophyll A here. That's why chlorophyll A is the main pigment molecule. So they all get transferred to chlorophyll A. What happens to the electrons in chlorophyll A? Just remember what happens to them? They get excited. And they get excited and they don't drop back down and go back to chlorophyll A. If that happens, remember I talked about how chlorophyll A will illuminate red. Remember that? All right. So these are two electrons that get excited, and the primary electron acceptor grabs them, all right, and doesn't allow them to go back down. That's what happens in the photosystem. So the electrons become excited. That's exactly how that happens. So let's look at then the bigger picture about what happens in the light reactions and where these photosystems are. Do you guys remember where they are? Where the light reactions occur?
Membrane.
Of what?
Thylakoid.
The thylakoid, yes. And so let's go ahead and draw that. So here's my big chloroplast. Like, I know that's a chloroplast, but if you're not going to remember that, you might want to label that. And then the thylakoid. Now normally there are lots of thylakoids stacked up in the granite and so on. But because I want to draw something and draw what's going on in the light reactions, I'm going to make my thylakoid really big in here. So this is one thylakoid. Again, it's not to scale, so it would be much, much smaller. But I want to show you that this is where, in that membrane of the thylakoid, so label that thylakoid. Let's just go ahead and label this the chloroplast. Alright, so this right here is your first photosystem, but it's photosystem 2 because they were numbered or named in the order in which they were discovered. And here is photosystem 1. Now this is why I went over this photosystem, because I'm not going to go into detail here about what happens. So now I'm assuming that you understand and remember what happens in a photosystem up here. And so, so this is where they're at. And then there's other molecules. You do not need to know the names of these molecules, but these guys form the electron transport chain, ETC. And so, so pulling in what we talked about here as a review, here are your two photosystems. There's pigments in there, and so light energy comes in. So here's my light energy coming in. Goes into both and absorbed by pigments in both photosystems simultaneously. It's going on at the same time. excited. And so then they jump the electrons, so I'm gonna put here 2 electrons, get excited, they go to the primary electron acceptor. I'm not gonna try and fit all that in here, because we just talked about it here. So therefore, I'm just writing that the electrons get excited, both of them simultaneously. So now this chlorophyll A, let's look at the chlorophyll A in photosystem 1 here. This chlorophyll A is now missing 2 electrons. These electrons have to be replaced. You guys remember where the electrons come from to replace it?
Water.
Well, ultimately from water, but directly right from here, these electrons go through the electron transport chain and replace those 2 electrons here. Let's go back to photosystem 2. This chlorophyll A had 2 electrons that got excited, so these 2 are missing 2 electrons. This chlorophyll A is missing 2 electrons. What directly replaces those? That's where water comes in. So H2O here. So the H's, the electron part of the H's go to replace the 2 electrons here. So these, I'm going to put 2 electrons, that's just 2 electrons. So it goes to the chlorophyll A molecule. That means that the H's now, we have 2 H pluses left over because they got rid of their electrons and gave them to chlorophyll A. And then the oxygen's not used at all here in the light reactions. What's the oxygen go? That's the oxygen that is released as O2 in the atmosphere. So that's the, when we say that oxygen is a product of photosynthesis, the oxygen comes from the water, right? And the electrons go to chlorophyll A. So then the only other electrons we have to account for is the electrons here that got excited. What are these? They don't stay with this primary electron acceptor here. What eventually gets these electrons? It's an electron carrier called NADP+. Alright, these electrons come over here and this becomes NAD. And remember that this is one of the big things that we want to get out of the light reactions is NADPH, like we wrote over here. Light reactions making ATP and NADPH. So we just made NADPH. What we haven't made is ATP. What did I miss when these electrons go through the electron transport chain? They do so because each one is more electronegative than the next. And just like in cellular respiration, when the electrons go through the electron transport chain, it releases energy to pump what through?
H plus.
H pluses. So out here, there's a low concentration of H pluses. And in here, inside the thylakoid, is a high concentration of H pluses. This is why I'm using the word pump, because it needs energy to happen. So when these electrons go through the electron transport chain, it pumps the H pluses into the thylakoid space here. And so now you've got this really high concentration of H pluses inside the thylakoid space. Why is that important? Because we want these H pluses to diffuse through, what's another molecule that's not part of the, here, it's a different molecule that the H pluses will diffuse through? ATP synthase. So this is ATP synthase here. H pluses will diffuse from high to low concentration. That changes the shape of the ATP synthase, allowing ADP and the phosphate to get into the active site and be converted to ATP. And ATP here is the second thing that is needed to be made in the light reaction. These guys here will go to the Calvin cycle, which is the next one. The only thing, so before we go to the Calvin cycle, there's one other thing I want to write on here. So this is the light reactions in a nutshell here. I just want to point out one other thing, and that is cyclic versus non-cyclic flow. I just drew a non-cyclic flow of electrons. Non-cyclic flow means the electrons are flowing linearly, so they go from one thing to another. And in non-cyclic flow, you make both ATP and NADPH. But if you recall, when I say that these guys go to the Calvin cycle, they're made in a 1-to-1 ratio, but they're not used in a 1-to-1 ratio. And so therefore, we go through what's called cyclic electron flow. Anybody remember which one is used more in the Calvin cycle? make ATP and no NADPH so that you can catch up and make with your ATP. So what does this mean, cyclic? Instead of these, if we look at photosystem 2, instead of these 2 electrons going to NADPH, because notice in cyclic we don't make NADPH. So what happens if I just cover this part up? These 2 electrons, instead of going to NADPH, they get dropped off in the electron transport chain and they cycle. That's the name cyclic. So they go round and around like this, meaning that the electrons continually go through the electron transport chain, which makes more and more H pluses be pumped in, which means that you can make more ATP and therefore catch up, because they're used in unequal amounts. Alright, and the last thing with photosynthesis is the Calvin cycle. Oops. 10, 3. Not supposed to be able to look there. This is its own entity, Calvin cycle. Where does the Calvin cycle take place in the chloroplast? Back to this picture, I said these guys go to the Calvin cycle. This is where it takes place. What is this area? Stroma. So it occurs in the stroma. So let's do another big picture here. Here's your chloroplast. This time I want to illustrate and talk about what happens in the Calvin cycle. So I'm going to minimize the light reactions here. So here's your little thylakoid with the light reactions. But I want to put them in here to bridge, and show you the relationship between what we just went over. And remember, the light reactions, 2 things are made, ATP and what? NADPH. And these guys are used over here, and we'll call this over here the Calvin cycle. Let's see in there. Calvin cycle. So the Calvin cycle is a series of reactions of which you don't need to know all these intermediates. So the ATP and the NADPH are used here in the Calvin cycle. That was the purpose of the light reaction, so that we can get the Calvin cycle going on here. So what enters the Calvin cycle? One of the reactants actually enters here. What is it? CO2. So I'm going to put 3 CO2. They enter one at a time, but 3 CO2 enter. This stage where the CO2 react with molecule X here, you don't need to know the names of it. It reacts here and makes, let's say, molecule Y here. That incorporation of the carbon into an organic molecule, this is the first step of the Calvin cycle. It's called carbon fixation. We had a lot of the questions today about carbon fixation. Carbon fixation is just a term that means the carbon and carbon dioxide now just reacts with another molecule, and now that carbon atom is part of an organic molecule. That's all that means. Alright, so it's no longer part of CO2, it's part of an organic molecule. And so then what happens is it goes through a series of reactions that uses NADPH. And so the second part of the Calvin cycle is called reduction. And in talking about, they call that in terms of electrons. And so the reduction, NADPH gives electrons to the molecules in the Calvin cycle. So the molecules in the Calvin cycle become reduced, hence the name reduction. So it goes through a series of reactions. It gives the electrons, those electrons help those reactions to happen. And then coming out of the Calvin cycle right here, you get your first 3 carbon molecule. You get 1G3P. This is a 3 carbon molecule. And glucose is 2 of these put together. So I'm going to put here times 2 will give you a glucose molecule. So then so right here there were 6 G3Ps. One of them comes out. The other 5 go through a series of reactions to regenerate molecule X. So like I said, you don't need to know what molecule X is. So the third stage is called regeneration. This part here is just to regenerate that molecule, so the next CO2 can come in and be fixed. Alright, so that's the regeneration. And so, then, just to add on here, when the ATP and the NADPHs are used, the ATP breaks down into ADP and the phosphate, and those can be reused in light reactions. The light reactions will regenerate ATP, so ATP is constantly cycled around here. When NADPH drops off its electrons here, what does it become? NAD may be P plus. Alright? And that's photosynthesis. In a nutshell. Any questions? Before we move on, 'cause I want to be able to get. Alright, let's go on. Moving on to chapter 11 then. So I'm going to do the same thing with the sections, 11.1. This was all about cell signaling. And so remember that we talked about local versus long distance signaling. Talked about it in the notes. We also did a packet that we went over the answers the other day about. And so endocrine, paracrine, all of those. So review those. And then there were the stages of cell signaling. Or the 3 stages. Reception, transduction, and response. So what we're going to do is the next three parts that we're going to look at here are these 3 scenarios. So let's look at reception first. So section 11.2 is all about reception. When the ligand binds to the signal molecule, or the receptor, so the ligand, what happens to the receptor? It changes shape, so that's what I want you to know. Binds to the receptor which changes shape. You need to understand that. Alright, so shape determines function. What's often changes shape. And we went through and I talked about 3 examples of this happening. So just want to examples of reception. So, and these are in your notes. There was the G protein linked receptor. So again, a ligand binds and you get a shape change. Just these different types happen a little bit differently. So then you have the tyrosine kinase receptors. And you have the gated ion channels. All of these are examples of reception. So review those. And that's reception. So then we get the second stage, which is transduction. We call this often when transduction occurs, we call it a signal transduction pathway. And sometimes within that, you get what's called a phosphorylation cascade. So I'll review that. That means molecules are phosphorylated. So let's go over that. So let's say I'm going to draw, this is the membrane of a cell. So this is outside the cell, this is the inside the cell. So let me just illustrate. Here's your receptor. So your receptor, here's your ligand binding to the receptor. So the receptor changes shape. So that's reception. So then let's look at how that relates to the signal transduction pathway and this phosphorylation cascade. Now when this receptor changes shape, it becomes activated. And it usually then activates another molecule called a relay protein. This activated molecule often will activate other enzymes within the cell. And the class of enzymes that's often activated are protein kinases. Do you guys remember a kinase? What in general do they do? They add phosphates to things. Alright, so therefore, let's say this is the activated relay protein. So what this activated relay protein is going to do next, notice I have an arrow here, it's going to take an inactive, I'm just going to put PK for protein kinase. If you don't know what that means, spell it out yourself. Alright, so inactive to active. So this molecule activates this. And this is an enzyme. So we have an active enzyme here. This protein kinase phosphorylates things, and it's an enzyme. So therefore, this active protein kinase, I'm going to draw another arrow arrow here like this. Notice I put the enzyme on the arrow to show that this enzyme is going to catalyze this reaction, and it's going to take inactive protein kinase, we'll say 2, and make that active protein kinase 2. So now we have this active enzyme. And how does it activate it? This enzyme is a kinase, so it activates other molecules by adding a phosphate to it. So I'm going to put here, off of this enzyme, a phosphate attached to it. Where did that phosphate come from? ATP. So also in this reaction, what goes in and goes out here for this reaction to happen and this to become active is ATP is used. And we take the phosphate off the ATP and put it on this enzyme. That means we have what left over? ADP. So it used an ATP to activate this enzyme. And then this enzyme could activate another enzyme and so on. And so that's the phosphorylation cascade. At the end of the phosphorylation cascade, you end up with an enzyme that's active, and it usually activates not a kinase, but another molecule. So I'm going to put another enzyme. So let's say inactive enzyme, random enzyme A here. So it's inactive. This last thing, the protein kinase, is going to activate enzyme A. So it's going to activate it to active enzyme A. And why would it be active? Because the kinase is going to add a phosphate to it. So here's your phosphate attached. To add the phosphate, it had to use ATP, right? So I'm going to put here, when we use ATP, we take off the phosphate and we end up with ADP. And now this enzyme at the very end is active, and this enzyme is going to do something in the cell that's going to lead to your response. So this is called the single transduction pathway, and part of that pathway is that phosphorylation cascade. Alright, so that is transduction. Now, when we look at transduction, so this is still under transduction. I'm just going to go to another piece of paper. I showed you every enzyme activating one other enzyme. Is that usually what happens? No, the signal is often amplified. So I'm going to put here many steps allow the signal to be amplified. What does that mean? So your relay protein doesn't activate just one enzyme. It activates many enzymes. And then each one of those activate many. And so on and so forth, so that at the end you get a big response from one signal molecule. Right? Does that make sense? So we could say, back on this picture here, this enzyme, instead of activating one protein kinase A, this enzyme can activate 100 of them, let's say. And then each one of those 100 activate 100. So then, and so on and so forth, so that you get that a lot, at the end, a lot of these active enzymes, which means you get a good response at the end from one single ligand. Okay? Alright, and the last thing underneath here is the role of second messengers. These are messengers that are after the ligand. And so I use the example of calcium ions and also cyclic AMP. So I'm just going to have you review those as second messengers. So I gave you some examples of that. Alrighty, and then the last part of cell communication is the response. So the response happens after the signal transduction pathway. I want to talk a little minute about where the response can occur. So the response can occur in the cytoplasm. So pulling in what we just talked about, this is your cell. Here's your receptor. So the ligand binds to it, that's the reception, and then you have the signal transduction pathway, and that response can happen right here in the cytoplasm. So it can activate an enzyme that's going to have a reaction happen right here in the cytoplasm. Another example of a response in the cytoplasm is sometimes the ligand actually goes into the cell and the receptor is inside the cell. What kind of ligand is this true about?
Oh.
What kind of ligand can get through the lipid bilayer? Nonpolar. So example of these kind are steroids receptor changes its shape, you get your signal transduction pathway, and then instead of the response happening in the cytoplasm, here's your nucleus, here's your DNA that has a gene as a segment of that DNA. That molecule enters the nucleus and comes in and it turns on a gene. And that's the response. So then that gene makes a protein and therefore you get a certain response in the cell. So that is another response turning on a gene. And the same thing can happen with the nonpolar. So just to relate this here, here's the receptor inside it. Nonpolar molecules can come in, bind to the receptor inside, same kind of thing. You get the pathway, and then it goes into the nucleus and turns on a gene. Right? And so that can happen in both ways. So you get the cellular response. When does the response stop? What makes the response? There are two things that we talked about that could make the response stop. We could go back to this picture here that we drew. So what could go on here to stop the response? We could deactivate these proteins. Alright, so how do we activate these proteins to begin with? Adding a phosphate, right? So we can deactivate them by taking alpha phosphate. So we can deactivate enzymes by removing the phosphate. There was an enzyme, by removing the phosphate. There's an enzyme that does that. It was protein phosphatases. In a packet we did, you had to add them onto the picture labeled with a P. Alright, remember that? The second thing that could stop a response, going back to this picture, is if if the ligand detaches. So the ligand detaches and these are deactivated. Because if you deactivate this and the ligand still stays attached, it's going to reactivate it and trigger this to happen. Alright, again. So the ligand detaches. And that's your response. The last thing that we talked about in chapter 11 in a we do is apoptosis. Remember that's a program cell death. And that's chapter 11. So you have the packets, the review packets for chapter 10 and 11. Those are due on Monday. I did send you the answers to those, so you have that. And then also, I do have some questions on chapter 45. I'm not going to do a review packet. I'm going to use the notes in this packet here.